And today we're doing a follow-up experiment from the last one. So, what preparation work did you have to do before this experiment?
I put a flower pot over a plant.
Would you like to just elaborate a bit further?
Yeah, a black flower pot over a plant with leaves, and then I blocked up the holes.
So, what was the purpose of doing that?
To,
Well, start with light.
yeah, light.
Light. Okay, so, can you pop the two leaves that we've got, because I've taken one off a plant that has grown in the light, which I've marked with an L, one off a plant which has grown in the dark, which I've marked with a D.
Yeah.
You use a biro for that. It damages the plant leaf just enough that you can see it when you get it back out again. Okay, and we're going to boil those. So pop the lid on the pan and turn the hob on. So we're going to boil these for about 5-10 minutes or so. No less than 5. You don't need to do it for much more than 10. And once they have boiled, then we'll carry on with the rest of the experiment.
Yes.
So, just before we go to the rest of the experiment, would you like to remind us all why it is that we're boiling the leaves?
Soften up the walls which have the...
Okay, so, yeah, that's right. So we're going to soften up the cell walls of the plant because they're made of cellulose and they're quite robust. They won't let the iodine in. The iodine solution is to test for starch, isn't it? Just like on the last video. So we're going to test for the starch, the presence of starch in the leaf using iodine solution. We need to get the iodine solution inside the cells, and in order to get it inside the cells we need to damage the cell wall first, so we're doing that by boiling it. In addition, the boiling it removes the outer waxy cuticle off the leaf,
Yeah.
which acts as a robust protector against the iodine gaining entry. Okay, and we will cut there.
Yeah.
And now we've boiled the leaves, so we're just going to flick them out carefully using our famous stirring implement and pop them into these two little shot glasses that we've put a small amount of alcohol into. So they go into there to soak. The soaking in alcohol will remove the green colour. Okay, I'm going to pop the lid on to keep them warm, and they're now in the hot water. Let's cut. So we've got the two little shot glasses with a small amount of alcohol in, and the leaves are in there, and the next thing to do is to whip the leaves out very carefully, and they should be very, very pale green, getting towards colourless.
Yeah.
Okay, and we're going to drop them in the hot water. There we go. Now, we've allowed ours to cool down quite a bit, so I can lift mine out. Yeah, okay, there's a chicken grooming herself over there. Just pretend that's not happening, you know. Let's just carry on. So what do we do now?
We take them out of the water.
Okay, so now they've softened up a little bit. We handle them very gently and spread them out onto our white plate. And do you want to spread that last one out whilst I get rid of the hot water and the alcohol? Here's the close-up. So what we're looking for now is for the iodine solution to cure on the one that's been in the light, and for it not to cure and to stay brown in the one that's been in the dark.
Doing quite a good job at that.
So why did we do the leaf kept in the light as well as the one kept in the dark?
So you could see the difference without watching the other video.
Yep, that would be one. So it acts as a control. Another one is there might be something wrong with the iodine solution. I have had that. I've worked in schools where our iodine solution has been far from satisfactory, and none of the leaves, even the ones kept in the light, actually turned black. So we've got a very, very nice clear curing there, so you can very easily see which one was kept in the light.
Yeah.
So which one was kept in the light? That's right. And the other one was put in the dark. So very briefly, and I'll stop there on that one, very briefly for the main camera, what we need to do is we need to say what we think is going on. We're just going to summarise quickly. The leaf that was kept in the light could freely photosynthesize. That's the light reaction. And it happens just in the light. The leaf that was kept in the dark could not do the light reaction for a 24-hour period. It was only in the dark for 24 hours. So it used its reserves of starch, it uses reserves of starch up to try to do all the other things that it needs to do. Now, what we normally say is there's also a dark reaction that takes place. It's part of photosynthesis, and the dark reaction takes place in the light and in the dark. And that's when the plant turns the glucose that it makes immediately from photosynthesis into starch. And then all the other processes which take place, where it breaks down the starch and makes use of it, and all of the other processes, they are fueled by respiration, which takes place in the light and in the dark. And that's why it had no starch left, because it had made 1 day's worth of starch for the plant, and then it had used it all up. The following day it used all the starch up still, and it now doesn't have any starch reserves. So now we have taken the pot off it, uncovered it completely, and allowed it to grow again. And that's that. Yeah. See you next time.